Hi, I'm Jenna. I'm a final year PhD student at Imperial College London, where I study how changes to DNA structure may be important for the evolution of ovarian cancer cells. So I started learning about cancer in my undergraduate degree, and I was immediately blown away by the scale of the problem. Because cancer cells, they're not just stagnant, but they're masters at evolving, at adaptation. They can evolve drug resistance, or they can simply become dormant until it's safe to reawaken. That means that for certain cancers, like ovarian cancer, over the last 20 years or so, the survival rates have barely increased, especially for late stages of the disease. So previously, a lot of work in the cancer field was based on this notion that cancer cells are caused by DNA mutations, which cause cells to grow uncontrollably. But now we're realizing that DNA mutations alone cannot explain why some cells develop cancers and others don't. So that's what led me to the world of epigenetics, of looking at how changes to gene expression may be key for cancer initiation and cancer evolution. The thing that I find so exciting about epigenetics is that epigenetic states are dynamic and they're reversible. And this is evidenced by the discovery of Yamanaka factors, where here you can take a few transcription factors and use them to reprogram cells back into a younger epigenetic state. And so the question then becomes, can we apply the same approaches of epigenetic reprogramming to cancer? So in my PhD, I'm looking at how epigenetic regulation changes as ovarian cancer cells develop drug resistance. I'm interested in how changes to DNA and RNA structure influence the transcriptional state of a cell. The structures that I'm most interested in are called G-quadruplexes, also known as G4s. And G-quadruplexes form through non-canonical hydrogen bonding between quartets of guanine bases. And this results in these 3D structures that are actually quite different to traditional duplex DNA. And the great thing about G-quadruplexes is because their structure is so unique, it makes it possible to design small molecules that will selectively interact with quadruplexes over other nucleic acid structures. And that means they may be ideal targets for modulating epigenetics that occurs within cancer cells. So in my PhD, I use methods like genomics and transcriptomics to map DNA and gene expression, but I also synthesize these small molecule fluorescent probes, which allow us to light up different DNA structures and image them by advanced microscopy techniques. But really, we are just at the beginning of understanding all of these epigenetic mechanisms that underpin the cancer world, and there's a lot more fundamental understanding that needs to be established. And that is what I am very excited to spend the rest of my scientific career doing.